Welcome to STEM Talk.
STEM Talk. STEM Talk. STEM Talk. STEM Talk. STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society.
Hi, I'm your host, Dawn Cornegas, and joining me to introduce today's podcast is a man behind the curtain, Dr. Ken Ford, IHMC's founder and chairman of the double secret selection committee that selects all the guests who appear on STEM Talk.
Hi, Don. Great to be here. Today, we have Dr. Marina Walther-Antonio, an assistant professor at the Mayo Clinic in Rochester, Minnesota. Her research focuses on the human microbiome in women's health, in particular, gynecologic cancers.
So we talked to Marina about how she and her colleagues are utilizing the methodologies of environmental microbiology as well as technologies used in astrobiology to improve our understanding of diseases such as endometrial and ovarian cancers. And through her investigations into the microbiome, she and her team have been developing early detection tests that will enable clinical interventions before certain cancers develop.
Before we jump into our conversation with Marina, we have a little bit of housekeeping. First, a big thank you to everyone who has subscribed to STEM Talk, and extra thanks for all of you who have left those wonderful 5-star reviews. If you're new to the show and you like what you hear, please consider subscribing. And if you enjoyed today's episode, we'd love for you to take a moment to rate the show and maybe even leave us a review. It really helps us reach more people who might enjoy STEM Talk.
As always, the Double Secret Selection Committee has been hard at work scouring iTunes and other podcast apps for the sharpest, funniest, and most over-the-top reviews to share on STEM Talk. If yours makes the cut and you hear it on the show, send us a note at stemtalk@ihmc.us to claim your very own official STEM Talk t-shirt, proof that the committee does, in fact, exist.
Today, our winning review comes from a listener who goes by the moniker RadDoc410. The review is titled Outstanding, and it reads, I'm a Pensacola physician and had no idea of the exceptional level of science and technology happening in my own backyard. I stumbled across this podcast from another podcaster's recommendation and wish I had known sooner of its existence. I look forward to every episode's release and only wish they came more frequently. I can't say enough good things about this podcast. Keep up the good work.
Well, thank you so much, RadDoc410, and thank you to all of our other STEM Talk listeners who've helped STEM Talk become such a great success.
He must be a radiologist. Okay, and now on to today's interview with Dr. Marina Walther-Antonio.
STEM Talk. STEM Talk. STEM Talk. STEM Talk. Hi, welcome to STEM Talk. I'm your host, Dawn Cornegas, and joining us today is Marina Walther-Antonio. Marina, welcome to the podcast.
Thank you. Happy to be here.
And also joining us is Ken Ford.
Hello, Dawn, and hello, Marina. It's good to be here.
So, Marina, later in this interview, I'm going to ask you about your thoughts regarding the possibility of extraterrestrial life. So I suspect listeners are going to wonder, why are you asking an obstetrician and gynecologist about extraterrestrial life? But we read a story about you in Mayo magazine that wrote about your fascination with ET life. And I understand this started in middle school when you asked your father if there was life beyond our planet. Is that correct?
Correct. So my dad raised me in a unique way, I will say, I think, in my family. So I grew up in Portugal. And up until 1975, the country was under a dictatorship. And I was born, shortly after that, in the early 80s, and my entire family and I guess the whole country was kind of traumatized with this whole experience. And so my dad was very set on raising me in a way that I wouldn't be just believing everything people told me or just following orders from others, and to be more of an independent thinker and kind of question things and have expression of thought. And he did this just with that intent of just wanting, his kid to be free. But inadvertently, I think he ended up raising a scientist because I would ask questions as any little kid does, and he would never answer any of my questions. He would always say, I'm not going to give you the answer because if I tell you the answer, you're just going to believe it because I'm your dad, and I don't want to have that kind of influence over you. I don't think I have the right to do that to you. So I can't give you the answer, but I can help you find the answer. So let's read about it. Let's find sources of information, and we'll come to a conclusion as to what the answer to that question is. So that's how it went. And it was a little bit frustrating sometimes because I just wanted the answer. But I think it was it was really an interesting way to make me very good at finding answers and searching for solutions to problems until I got to middle school, and I started asking about origin of life and where do we come from and what are we alone? And he basically told me, I don't think we have the answer to those questions, and that just didn't make sense to me. I was like, this seems like the most important question anybody can ask. How do we not know the answer to this? And so I really took it as a mission to try to find an answer to these questions. And I kind of started there, my interest for, understanding life on Earth and beyond, and what kind of people were studying those things, and how could I get involved in that. And it continued until this day, really. Yeah.
So, Marina, I think that's a great mission to pursue. And it's great that led you into science. So, you did your undergraduate studies in Portugal at the University of Aveiro, where you majored in biology. So, why did you choose biology?
Because it was one of the main things that I was interested in. I was interested in the scientific field in general. I did think about astronomy because that was another thing that would connect me to aliens, I guess. But I was really good at biology at that time. I also grew up in a farm, and I was very interested in animal behavior and just felt very connected to nature, and it was very intuitive to me how biology worked. And that was the kind of field of science I felt I was more interested in and better at. Not as great at physics and that sort of thing. So that's, that drove me to, that's going to be my element. It's going to be biology. Yeah.
As part of your undergraduate curriculum at Aveiro, you were required to do a year of research outside the university, and I think this is always a great idea. You applied for a number of internships, but in the back of your mind, you kept thinking you should do something kind of crazy, something different. Can you talk about that?
Yeah, so my astrobiology interest that I had kind of always been in the back of my mind, it was still there. And, I did think it was entirely crazy because, I wasn't a particularly great student. I was a good student, but not like a great student that could go anywhere I wanted to. That was not the situation I was in. And of course, the only place that was really doing astrobiology at that time was NASA, right? So and that was a crazy idea for me to think that, I'm in Portugal, and, kind of average student is going to have a shot at this. But I took a look and saw that they did have an internship open for, kind of undergraduates interested in astrobiology. And I thought, well, what do I got to lose, right? I'm going to apply, you're going to say no, and I move on, I guess. And at least I say I tried. And so I did apply. And Lisa Pratt, who was the director at the time of the IPTI Center within the NASA Astrobiology Institute. So she was one of the PIs in the Indiana, Princeton, Tennessee Astrobiology Initiative. She took a leap of faith, I guess, at the time and accepted me into the internship program there. And here I was. I hadn't even told my parents that I had applied for this because I didn't think there was a need to even, mention this. And then I was like, well, I got into this internship with NASA in the US, and it was kind of pretty wild, but yeah, it was the start of a great journey, I think. And I was supposed to come here to the US for about a year, and it's been over 20 years, and I'm still here. So it turned into a pretty long journey, but a good one.
Yeah, it had to be a great experience. And when you were there were quite a few different projects going on in the Astrobiology Institute, and you were plugged into a project that was looking at a Mars analog site in the Warner Valley of Oregon. What kind of work did you do as part of that project?
Yeah, most of the work I did. So Lisa Pratt is a geomicrobiologist, and she was looking at mostly into the geological setting of Warner Valley, which is an analog for early Earth and early Mars, as you mentioned. The hot spring systems, the alkaline salt flats as well. And I was with a biology background. The collaborator she had was Carl Bauer, which was at the time the chair of biochemistry at the Indiana University, and interested in kind of starting to look into, it was mostly working in biochemistry, but he was interested in looking in what was relatively new at that time, since 20 years ago, mind you, I'm old. So, but the molecular approaches, kind of 16S, ribosomal DNA, and cataloging microbial life. So I kind of went in that direction to go to those sites, and along with studying the chemistry and the geology of these places, to study what kind of microorganisms could we find, in Anderson Lake and the hot springs surrounding that and alkaline salt flats. And we started kind of cataloging all that in what we can classify as environmental microbiology approach, but with a thought to inform future missions to Mars on what kind of things we could expect to find in those kinds of environments.
So after graduating from Aviero, you enrolled at Indiana University to major in microbiology. And then after you completed the master's degree, you headed to Washington State University for your PhD in environmental sciences. So how did that transition work exactly? A microbiologist decided to make the leap into environmental sciences. Could you talk a little bit about that?
Yeah, because it's a pretty wild story, actually. So I was in Indiana University. I'm kind of a person, because probably of my dad, I guess. I have strong opinions sometimes about something if I do my research and kind of think I know what I need. And so the programs at Indiana University, they're very stiff, I'll say, in their structure, in the sense that they had, microbiology kind of degrees have to have certain courses in certain directions. And I was more interested in exploring evolutionary side of things than the biochemistry side of things, just based on what I wanted to interrogate. And the program wasn't very flexible to give me credit for those courses or, giving me that flexibility. So I was a little upset about that. And at that time, I went to a conference in California, an astrobiology conference. And I met Dirk Schulte-Makuk, which became my PI at Washington State. I gave a talk there. He's a very unique individual, eccentric, I will say. Very novel, crazy ideas for a lot of people. And I thought that was really refreshing. I really enjoyed his talk, and I had my own talk too. And he also liked my talk. We kind of connected at the conference. And I remember he came to me and he said, when, doing your studies in Indiana, and I had initiated the first year of PhD at this point in Indiana. And he said, when are you finishing your PhD? Because I would love to have like a postdoc like you at Washington State. And I said, oh, I'm just in my first year. I just finished my master's now. I'm moving to a PhD. And he's like, oh, that's really unfortunate. And, we kind of, went our ways. And this was, I remember, like a Friday or something. And by the time I got back to Indiana, I checked my email on Monday. I had an offer to transfer to Washington State. So he had gone back to Washington State and convinced all the people there that this must happen and I must go there and do a PhD there instead. And, talked to Carl in Indiana about this, went to visit, and he was okay with that. And so we, we ended up going in that direction. And the program that allowed me the flexibility I needed and to do what I needed to do was the environmental sciences and natural resources, which allowed me a lot of flexibility in the curriculum. And then I talked to the program directors there, and it really felt like this was a great match for what I wanted to do and explore more of the astrobiology side of things, which is in between disciplines. It's not as connected to a specific discipline, but it's transdisciplinary. And I needed a program that understood that. So that was, I think, a great match in terms of program and also mentor. So that's how it happened, which is kind of crazy, but it's a true story.
Very interesting. And a lot of people's life stories, when we interview scientists and engineers, they're not straight lines. They're nothing that could have been predicted in advance. I think that's a good thing, really.
Yeah, most of the most interesting people we know never drive in a straight line, or at least in life. So yeah, absolutely. I love that story. So as part of your PhD research, you analyzed microbiolites, which are microbial structures that can thrive at the bottom of certain freshwater lakes and other extreme environments. So during this research, you had a kind of aha realization, didn't you?
Yeah, so one of the systems that we studied then at Washington State was the Pavilion Lake in Canada. These are freshwater lakes that are mostly frozen during the winter, and they create there's these structures that are emerge from the bottom of these lakes that are similar to stromatolites. I don't know if people are familiar with those, but they are typically found in Australia and more saltwater type of environments. One of the first life forms on our planet, and these microbiolites are similar to those, but they're freshwater and they're kind of greater depths. And we were interested in studying this as an analog to Europa, for example, Jupiter moons, and trying to understand what could be lurking under layers of ice, basically in those environments. And when we started looking at these structures, they're several meters high, but they are entirely built by unicellular organisms from the three domains of life: archaea, bacteria, and eukarya. And it was very interesting because they form these structures that appear to be very coordinated. And so we were kind of interested in understanding how is it that they are communicating with each other to do this. Because again, these are from the 3 different domains of life, and they somehow are coordinating these activities. And when I started looking into the molecules that they were utilizing to communicate, I started hitting a lot of cancer literature, which did not make sense in my head at the time. And I was like, what, why is this where I'm finding these molecules? And it turned out, to lead me to think that, if we have these kinds of microorganisms or similar things in our body, and there's these cancer researchers trying to use these pathways to treat patients or understand what's happening, and they don't know that these microorganisms are there fiddling with these same pathways, that could lead to a lot of problems or a lot of misinterpretation or results that just don't make sense, because they're not taking into account an important part of their ecosystem in a way, because our body is also an ecosystem. And so this was kind of at the time that the microbiome field was, or the human microbiome field was starting to emerge. And so people were starting to think about these things, and it really kind of made me pause and think about the possibilities that could be happening, and how important it was to look into that too.
It's really interesting. And so we've been interviewing scientists and engineers for STEM Talk for almost 10 years now. And one thing that we've learned is a lot of their success and careers have come from being just in the right place at the right time.
Yep.
That path that, isn't necessarily straight, but it makes sense that when you look at it in retrospect. So as fortune would have it, just as you became obsessed with microbial populations, the Mayo Clinic Center for Individualized Medicine created a microbiome program. So I understand 2 strange things happened that led you to heading off from Mayo to become a research fellow with the microbiome program. Is that correct? And can you expand on that a little bit?
Yeah. So, going back to that conference in California that led to my transfer from Indiana University to Washington State University, at that same exact conference, which was a Gordon conference, I met Nicholas Chia, who was at the time a postdoc at the University of Illinois, Urbana-Champaign, a physicist that was working in astrobiology. And extremely bright guy, and we connected. So he was a postdoc, I was a graduate student at the time. We kind of connected at the conference and kept in touch, talking about, he always had this really novel ideas about what was going on in the biology side of things. And so we kept in touch, and he ended up at this time to be the first faculty to be hired into this Mayo microbiome program and to help initiate that program. And so he reached out to me and he said, "Hey, are you graduating soon?" Like, okay, he knew I was getting into my end of my 4th year in a PhD. And I was like, "Well, I'm getting close." And he's like, "Well, can you do it in a few months?" Because I got a position for you here. I'd really love you to join the team. Given all we've talked about, I think we have similar ideas and alignments on these things, and you would be the right person for this. And to help kind of transfer a lot of the knowledge we have from the environmental side and, pipelines and concepts and approaches to studying this in the human body and really looking into these questions. And, and then I talked to the director of the microbiome program that had been appointed, Heidi Nelson, and her pitch to me was quite convincing. She said, "Can you look for aliens in the human body?" And I was like, I could, So, and I said, as long as I can continue to look for aliens elsewhere, I think we have a deal. We'll do both. And she's like, "Deal. We'll do both." So that's how we got into that agreement, and that's still going. I've been at Mayo for 13 years now, and I still do both things, and I'm really happy that they let me do both things, and because they have been able to, I think, help each other. It's fields that you wouldn't think are or should be connected, but there's a lot of things that actually transfer very well, and insights that I get back and forth that are pretty unique, just because I happen to be there's not a lot of people that do both things, I guess. So I'm in between 2 worlds that not many people cross paths with. So it's been pretty interesting and fun.
Yeah, it's great to have that shared perspective. So we had Dr. Claire Fraser, who is the director of Maryland's Institute for Genome Sciences, on episode 32 of STEM Talk. And she pointed out that there are more microbes on a person's hand than there are people in the world. And she also said that our gut is home to more than 100 trillion bacteria. So I guess when we think about it, we're pretty much a bacterial organism, or at least a vessel for bacteria, aren't we?
Yeah, when you think about the numbers, if our body has more bacterial cells than human cells and more microbial genes than human genes, then I guess we're mostly not human, right? If you think about that. Yeah, it's very interesting to think about it in that way. we are never really, alone. That's what I always tell sometimes my students too. It's, if you feel lonely, just think there's millions of creatures with you right now. The fact that we haven't really thought about it or take it into account into a lot of the things we do, I think hasn't been great. I believe that, ignoring it doesn't make it go away. And what I always tell other scientists sometimes, it's that, that are doing, for example, maybe genomics or something to understand disease. And I know I bring this up, what about the microbiome? And they're like, I think we have enough of a problem to solve here with just genetics and all that. And I said, well, yeah, but like Einstein said, you should, everything should be made as simple as possible, but not simpler, right? That is the biggest mistake anybody can do, because then you really are missing a part of the problem that you'll never be able to solve if you don't take into account everything. So I think it's always better to assume that everything could have a role and then, eliminate that's a contributor to that. That's fine. You can determine that this doesn't seem very relevant. Okay, we can move on. But if you never even look at it in that direction, you could be missing the key right there. The needle you're looking in the haystack could be right there, and you won't have it in your pile if you don't think about it. And given the numbers that we just mentioned, and the fact that microbes are with us from birth to death, and are able to metabolize things that we can't, and are able to transform things and drugs that we take, should really give everybody pause to just think about what else could be happening with your patients if you're a physician or whatever you're trying to solve. Right?
Mm-hmm. I can appreciate how many find the computational complexity of the microbiome somewhat daunting, and so they sort of stick to their knitting. And it's so true what you said. I think it's absolutely the case. And, we tend to not look at things that are beyond our ability to currently handle them effectively. The individual variation is great and the complexity is great. But as you say, not looking at something like that seems quite unwise, and I'm glad you're doing it. For listeners who are interested in learning more about the microbiome and how it affects our health, there's a couple episodes, also earlier episodes I could point you to. One is episode 20 with Dr. Alicio Fasano, and another would be episode 168, also with Alicio Fasano. And these are both quite interesting episodes. Now, in terms of research, as we were just discussing, it seems like there's still much we don't know, though in recent years, the research into this area has really exploded. So, Marina, before we go any further, could you give our listeners a short intro, sort of a microbiome 101, and then we'll jump into your research?
Yeah. So the microbiome field, and I'll define it in perhaps in comparison to what is more familiar to most people, which is the microbiology field. And the microbiology field is somewhat split between environmental and clinical microbiology. And, environmental microbiology is studying microbes in the environment. Clinical microbiology, more things related to disease and pathogens, typically. The microbiome field kind of emerged of this idea that, there are microbes that exist in our body that aren't pathogens. They may, in fact, be beneficial. I sometimes think of it, if people, maybe some people are familiar with the symbiosis that exists between, rhizobium and some plants and that attach to the roots. Some plants aren't able to fix their nitrogen, right, to grow. And they aren't able to exist without these rhizobium species that actually do this fixation of the nitrogen. And that's how they've evolved to depend on each other, basically. Rhizobium also gets the sugars and things that they need from the roots. So this kind of symbiosis, is clearly present in most organisms, and humans are no exception to this. We have them in our gut. A lot of microbes are able to help with the absorption of macronutrients that are needed. So what I often tell other people is when you think about microbes in our body, think about, that we often think, having a particular microorganism present or a pathogen is a problem, right? It's a germ, it's an issue. But think about what could be an even bigger issue if microbes that you actually need for your survival and for your well-being and for your nutritional balance aren't present. That's a much bigger problem, can become a much bigger problem to solve, because now you're really missing a component of your body that we as a species have evolved to depend on. We depend on the presence of these microorganisms to be healthy. And if they disappear for some reason, it could be because antibiotic treatments that were needed for some other reason, but, it can offset the balance. It could be that we're not ingesting what we need to sustain those microbes through our diet. We can lose them. And getting them back isn't trivial at all. It can be quite challenging sometimes. A lot of what I think is particularly complex and chronic diseases is where we start to think more about this. Could be, also a result of some of those imbalances in the microbiome where you're just missing some components that your body needs, and that for some reason either you didn't get them or you lost them or it is weakened, and we need to restore that balance. So I always kind of see the human microbiome as a field in between environmental microbiology and clinical microbiology, in the sense that we look at the human body and the microorganisms as an ecosystem, and how do we get that to a healthy balance and keep it there and build resilience in people so that when they have to go through harsh treatments for whatever reason, that they can maintain what's so important to for their overall health, to be able to withstand those treatments, to improve afterwards and recover. That's really important.
That's a fantastic overview, Marina. So, your current research focuses on the role of the microbiome in cancer and reproductive health. So, how did this become the focus of your research and your lab?
Yeah, so also not the typical way. I think a lot of people kind of have a very particular focus. I don't, obviously, because I'm all over the place. When I joined Mayo, Heidi Nelson, she was the first director of the microbiome program, as I mentioned, and also the chair of the Department of Surgery. She's retired now. But she said something that is I appreciated more every year, I will say this, because it's so rare. So she appointed me in the Department of Surgery because it was her department and it was easy for her to do. But she herself told me this, that just because you're in the Department of Surgery, don't feel constrained that you have to work with just people in the Department of Surgery and faculty in the department. What I want you to do is to go out. Mayo is a big place, or even external collaborators. I know you have international collaborators, which I do and still maintain. Go find your collaborators, go find your people, talk to folks about what the problems they have and what they want to solve, see what you're interested in doing and who you're interested in partnering with, and build those partnerships. Feel free to do it whatever it needs to be done. And that provided me a freedom that, I will say not a lot of places, a lot of people are afforded that. So I do appreciate it a lot. So I did just that, and I, connected a lot of folks, and I do have projects in many different areas. But I will say that the Department of Obstetrics and Gynecology, which I am now, it's actually my primary affiliation right now, there were a lot of individuals in that department that were really interested in this. And I think it's because, a lot of issues with women's health and gynecologic health, obstetric health, just haven't been researched as much as other fields. And so there was a lot of doctors that felt they had an inclination to think that this had some microbial nature to it, but it was something that, hadn't been resolved by clinical microbiology or the typical approaches. And they really were very excited to try to see if something else could help them help their patients. So, it was the enthusiasm of these doctors. What I did with the starting conversations with physicians, often what I ask them when I meet with them the first time is, what keeps you awake at night? And they look funny at me sometimes, but what I mean, is what kind of patients, what kind of problems do you go to sleep and think, gosh, tomorrow I'm going to see this patient and I'm going to struggle to help them because I don't have solutions right now. Like, what is that kind of patient? And a lot of these physicians in the OBGYN department have an answer right there. They would be like, oh, this one, like tomorrow there's this patient, and it's been, 10 years we've been trying to help this patient because, this and that. And I knew right there, right, when people are that, like, have an answer right there, I know that it's something they're deeply motivated to solve and are personally invested in the issue, that they will make up the time to do it. Physicians are very busy, obviously. So, but if it's something that does keep them awake at night, they will be hell-bent on trying to solve, because it's really important to them. So that's how I kind of chose those areas that had those people that were that committed to their patients and solving their problems.
That's an excellent background. And maybe just digging in a little bit more here. So, endometrial and ovarian cancers are among the top 10 most prevalent cancers in women worldwide. And actually, ovarian cancer is the second most common gynecological malignancy and is also the fifth leading cause of death due to cancer in women in the US. And this is largely due to the late stage diagnosis of ovarian cancer. So, you investigated the microbiome that's associated with ovarian cancer to better understand the microbiome's potential in the early detection of the disease. So could you talk about your 2023 paper that reported on the insights that you gained from this study?
Yeah, and I can give just a little background. I think we'll talk about maybe endometrial cancer next, but the endometrial cancer project came first, and what happened was we had some promising results there that we'll talk about. But when I was giving a talk actually within Mayo about presenting some of these results, Scott Kaufman, who is the director of the ovarian cancer spore at Mayo, was in the audience, and he jumped up and said, "You must do this in ovarian cancer. This could be so important." And I was like, okay. So he was one of those people that was absolutely committed to for this to be done in this space. And so I have been working by now at this point for 3 years in endometrial cancer. And, I spent those 3 years. So when I moved to Mayo, just to give a little background, I obviously had no medical training or none of that, right? Because as I said, I come from the environmental field. And so when I landed here, I told Heidi Nilsson that I feel like I just landed in a new planet, which is medicine, right? And I need time to just figure out what your world and what you do, and so that I see how I can help. And so I spent these 3 years in the ORs, in the pathology labs, and kind of just understanding, I watched hundreds of surgeries and kind of how to sample things, when I could do things, when I couldn't, and what the best times were to do certain things that wouldn't disrupt the clinical work, surgical work. So I had seen a lot of, endometrial cancer surgeries and all those surgeries. And then when I went to enter the ovarian space, I remember the first ovarian cancer case that I went to watch was the one that really shook me quite a bit, because it was what's called an open and closed case, which I hadn't seen in endometrial cancer, even though I've had seen hundreds of these surgeries. And even the mood in the OR was so different that it kind of really hit me. All the surgical team are typically, in a good vibe, good mood, and this was not it. When I went to the ovarian cancer case, there was a very somber silence. I could tell the surgeon was, looking and see what he could do about the case, and it was it turned out to be a very advanced case that could not be surgically resected. And within 20 minutes of this incision, the surgeon said, it's an open and closed case, and I'll talk to the family, but we're going to move on to the next patient. And knowing the Mayo is a tertiary care center and all that, I was well aware, there was still hope for that patient in terms of, doing first some adjuvant treatment. But it's a very, you realize the seriousness of that cancer, right? And how devastating it is to be even for the surgical teams to not be able to do that, because if we can't do that at Mayo, then, we know that's kind of the end of the line. So that really was very eye-opening. And then the statistics you say, yes, but of course, being in the ORs and all that, you get to kind of experience that firsthand for each person. Each one of those numbers is a person, and there's a family, and there's often these women that get diagnosed with these cancers are young moms, and it's just very devastating. So I became very committed to try to help in this space. Of course, it's very challenging because there's no risk factors other than genetic BRCA1 mutations, that sort of thing, or hereditary, but there's no early symptoms either. So it's a very difficult space to work on in terms of prevention or early detection. So, but nobody's had ever looked into this, and I knew there were a few groups starting to think about this at this point in time. And so we joined, and we started collecting some samples, similar study setting to the endometrial cancer, where we collected, all kinds of samples from the patients, so vaginal swabs and scrapes and cervical samples as well, in the uterus after it's extracted from the patient. So after hysterectomy, we take some tissue samples and also the fallopian tubes, the ovaries. We took ascites, which is a fluid that builds in some patients, abdominal fluid or peritoneal fluid, and stool samples, urine samples, just because we didn't know what kind of signals we could potentially find in terms of finding a microbiome that was associated with the disease. And what we used as controls was patients that were having a gynecologic surgery for some benign reason that wasn't ovarian cancer, but something else, so that we could see if any of the signatures we could identify in the microbiome were specific to ovarian cancer. And, what we found was that, yes, there's some signal. I mean, this cancer is not as common as others. So, we study is limited by its size. So it was about 30 ovarian cancer patients and about the same number in controls. And we did find a signal that a combination of microbes that was significant, but we need more numbers to be, for it to become kind of something that could be used clinically. It's not at that stage. So this was a discovery stage. What we did find, somewhat by accident in a way, because we didn't design it that way, but when we looked at the data and we're like, okay, there's some signal. It's it's kind of there's some signature linked to some combination of microbes, but clinically this would be difficult to think how to deliver this. But we did notice that patients who after the surgery responded to therapy versus those that did not, that signature was actually stronger and more promising for clinical use. So we are kind of following up on those results and thinking about that a little more and what we can do about it. Because right now, of course, there's not a lot of treatments that are very good for ovarian cancer. But if we can tell with some certainty that some patients will not be responsive to treatment, they can be fast-tracked into clinical trials or some other type of therapy earlier, so that we don't waste time and the health of the patient in trying something that it's likely to not work. So that's kind of how we are thinking and moving in that direction. Though the prevention side, we're still thinking through it, but it's just the signature is not as strong as what we found in endometrial cancer and that sort of thing.
That's really interesting. And just a minute ago, you pointed out that it was a relatively modest size study in terms of the number of participants, and that you thought it should be followed up by a larger study. In your mind, other than more participants, what else would that study involve, the one that you envision for the future following up on this study?
Yeah, for the prevention side, I think one of the things that is important is to also try to catch these cases as early as possible. So, and again, because the cancer is rare, this is not something trivial, but the point of ovarian cancer is to find it early, right? So to try to find patients that are early in their diagnosis is really important. But yeah, again, I did talk. I had this is something brought to me by a patient advocate that I thought was quite interesting, and we're thinking along those lines now too. But she had mentioned to me that she herself was in a situation where she has a family history of ovarian cancer, but she's not BRCA positive. So she's in this situation where, clinically, yes, there's a hereditary reason to think that you're at elevated risk for ovarian cancer, but it's not something that they can tie to a mutation or something like that. And so she said, there's a lot of people in my situation and we don't know what to do, because it's like, should I do prophylactic surgery basically and remove the ovaries, which is a big deal, of course, for her health. And, for a lot of reasons, that's not something you would do not for a very serious reason. So, but, there's this fear of, I am at increased risk given the family history, but it's not something that they can definitely say I will develop this. So if there was something that the microbiome or whatever could tell me, am I really at increased risk or am I not, or what should I do, right, to kind of guide that decision making for these patients. And that's something we are thinking to potentially to try to help with that, those kinds of situations.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research organization investigating a broad range of topics aimed at understanding and extending human cognition, locomotion, health span, resilience, and performance.
Back in 2019, you published the results of a study that found that postmenopause is a key factor in the composition of the endometrial cancer microbiome. This study also investigated the impact of a number of patient factors in the microbiome and how those factors might contribute to the microbiome's signature in the cancer itself. These patient factors included menopause status, body mass index, and vaginal pH. This was a very involved study that found each of the factors we just mentioned, especially postmenopausal status, do alter the microbiome associated with endometrial cancer. Can you talk about this study and the significance of your findings?
So the endometrial cancer study focused, a similar design to the ovarian cancer study, and it was the original one. What we were interested in trying to find out is, for endometrial cancer, there are risk factors that have been identified. And that's, obesity is a risk factor. Diabetes can be a contributor, and postmenopausal age is typically when the cancer is diagnosed. So we kind of did the study to try to understand, well, which of these factors is contributing or is a result of something else, and kind of try to figure out the chicken and the egg type of story. And yes, indeed, we found that while obesity does modify both the gut and the vaginal microbiome, as we know now, it doesn't modify it necessarily in the direction or enrich for the microbes that we see postmenopause enriching, that are then aligned with the endometrial cancer. And what we started thinking, and this is due mostly, I will say, to my environmental background, but kind of this hypothesis of ecological succession, right? Kind of this idea that, just like you have in ecology, you have, for example, a beach where, you can't just plant trees and they're not going to grow there. It's just not ready. The soil's not ready to support that kind of bigger organism. It's going to start with grass, and then the grass is kind of maybe going to transform the soil to be able to then later grow into bigger types of plants. Kind of a similar idea for this. So you could have, an environment that is maybe healthy at the beginning. Maybe you have some microorganisms that are opportunistic pathogens, which, everybody has, and, they're there and they're friends until they get an opportunity, or maybe they start struggling to find something. Our hypothesis was that this organism that is the most associated with endometrial cancer, which is Porphyromonas samurai, that this microorganism we know is a poor iron chelator. So in essence, it needs iron, but it's not very good at getting it. And, when women are in their reproductive kind of years, they have good irrigation in the uterus. There's a lot of available blood. There's menstruation. This microorganism has no good reason to start doing anything to get iron. Doesn't need to. It's right there. But if you still have this organism and then you enter menopause, and now suddenly that irrigation, that easy iron source of blood isn't around anymore, this microorganism, because it's not very good at getting the iron, is going to start struggling. And so at this point is what we start hypothesizing is it's going to try to find a source of iron. And the best source of iron, if you can't find it through circulation and in the environment, is inside the cells. And so Porphyromonas gingivalis, which is a cousin of this microorganism, so to speak, it causes periodontitis, does this in the mouth. It will invade the gums and gum cells and create that bleeding to get access to that iron. I call them kind of a vampire type of thing. And so this Porphyromonas samurai, we're hypothesizing may be similar to this. And one of the first signs of endometrial cancer is bleeding in menopause, right? And so that would be kind of the periodontitis, right? The bleeding gums, but in the uterus. So that's what we're thinking. But also as part of this succession, what we're thinking is that Porphyromonas samurai is not very good of a pathogen and kind of needs others to help out with disrupting some extracellular matrices and kind of being able to do some of these things. And so I think there's some succession into, going from a healthy microbiome to something that is permissive to the presence of Porphyromonas samurai, and then to it kind of helps it like astrobiome, like Prevotella and Peptonephelus types of microorganisms that then can facilitate this microbe to invade cells and do worse things. In the study that you were mentioning, we did find that there were other microbes associated with the disease. We also showed through a blind clinical test with just a vaginal swab that we could distinguish with an 86% positive predictive value, somebody who had endometrial cancer from people who didn't, just based on the presence of this microorganism. And then we had a follow-up study to kind of show that indeed it is able to invade the cells and go along this sort of hypothesis, and then what happens after it's inside the cell and so forth. We're trying to study kind of the mechanism involved and how we can stop it if that's the case, or how if it makes a difference. We're conducting right now, it's not published, but we're working on animal studies to see what exactly how is this microbe involved in this, if it's just associated with the early progression of the disease, or if it's actually causative and that sort of thing.
So here we are in 2025, and even though ovarian and endometrial cancer are among the top 25 most prevalent cancers in women worldwide, there's still no standard screenings for early detection. So this is something that you and your colleagues in your lab have been working on. Can you talk to us about the work that you've been doing in this space?
Yes. So for ovarian cancer, as I said, it's a very difficult space for screening because, again, our inability to have identified risk factors other than hereditary and also absence of early symptoms. So and the rarity of the cancer. So those three things make it a very difficult place for screenings unless the evidence is overwhelmingly strong for a particular marker, which I don't think anybody has been able to get there yet. For endometrial, it's actually quite different. Endometrial does have early signs with the postmenopausal bleeding. As I mentioned, that's a sign that requires checking. And also we do have some risk factors, but there is no early screening test for endometrial cancer. I will say primarily because, I would say 90% of the women that develop endometrial cancer manifest early with bleeding. And so that's considered, reasonable. Now, the main issue here is that the 10% of women that don't either develop bleeding or don't recognize it, or the physician doesn't recognize it as an issue, do develop, very serious disease that it's as difficult to treat as ovarian cancer is. And unfortunately, those women, the women who are minority women, so Black women in particular, have a higher prevalence of these types of cancers that are more aggressive and tend to not bleed early or have other confounding conditions such as fibroids, for example, which are also more prevalent in Black women, and those cause bleeding as well. And so they can be dismissed by the patient as attributing their bleeding to the fibroids. They can be dismissed by the physicians as well as for the same reason. And so this has caused a lot of health disparities in the field, which, if you look at the numbers, you'll see that the incidence of the cancer is similar between, all races and ethnicities, but the mortality and morbidity is far higher for Black women for a lot of these reasons. And so, what we did write, I think me and my clinical collaborator in this space, Andrea Mariani, wrote an opinion article at some point indicating that, if the rates of endometrial cancer morbidity for Black women were the same for everybody else, we would be talking about screening tests. But because they're only impacting mostly Black women, it doesn't seem to cause the same urgency to solve the problem. So, but this is something that a lot of people have identified as a very important issue that we do need to solve, and potentially a screening test would be justified to put an end to this disparity.
So, prior to this study, vaginal pH has not been associated as a risk factor for endometrial cancer, and variations in microbial composition are implicated in a number of gynecologic and obstetric diseases. As we routinely point out on STEM Talk, good science begins with good questions. And a question that you had was that the vaginal microbiome is so crucial to women's health. Why do vaginal microbial profiles vary strikingly from person to person and even change over time within the same person? And this question led to a study and a 2020 paper titled Daily Vaginal Microbiota Fluctuations Associated with Natural Hormonal Cycle, Contraceptives, Diet, and Exercise. So, Marina, can you give us an overview of the study and also its key findings?
Yeah, so this is an ongoing study. Still to this day, we published that, but there will be more coming. This is a collaborative study between us, Mayo Clinic, and Wellesley College, and Mark Tittel, our collaborator there. And Nicholas Chia, who was, the postdoc I mentioned earlier on that brought me to Mayo, who's now at Argonne National Labs and is also still working on the project. Our motivation here with this was precisely what you were mentioning, that, we all want to solve patient problems and health issues, but when you're starting a new field, it's the issue is that we don't even understand the basics or what's normal, right? We need to understand what is normal to understand what's abnormal. And there haven't been a lot of studies describing in detail what does the vaginal microbiome look like for everybody every day. Is it always the same, right? I mean, there have been some studies in the gut microbiome, but vaginal is really not as much. So we had a group of students at Wellesley that it's a student population that it's very research inclined and interested in helping out with this kind of research. And so we started this study with a long-term thing in mind. But basically what we did was to ask these students to collect vaginal swabs every day for a period of about 6 weeks. And not only that, but they're also, collecting stool samples when they have bowel movements. They have a Fitbit that tracks their levels of activity. They also have a saliva test to kind of track ovulation. They have an app on their phone where they are asked to eat while they're in the study, to eat at the cafeteria, where they can kind of select what meals they're having. So we get the exact nutritional information from the cafeteria directly on what they're consuming, so we can take that into account. And we also have a questionnaire that they can track every day. There's markers for anxiety, depression, sexual activity, hygiene practices, all kinds of things. And so we're trying to understand, what does the vaginal microbiome look like on a daily basis, right? Through all of this and understanding everything that's going on. So that study kind of looks into that. And what you'll see is very interesting to me. And I think it was very eye-opening to me, and I think for a lot of other folks, in that one of the main criticisms that people who look into the microbiome may have heard or even said themselves is that, well, it looks like every time you collect a sample, it looks different. What does that mean? How can you make sense of that? Right. And if you look at that data that we published in this paper, you will see that there's, daily fluctuations of the microbiome as they go through those 6 weeks of the study. But if you zoom out and you just look at each profile of each of those participants, I can guarantee you that you or me or really anybody, if I would give you a sample and asked you which participant is this from, you would have no problem identifying it, because yes, it changes every day, but it's somewhat predictable and it's pretty characteristic to that particular individual. So it's very interesting to kind of just realize that it's all about perspective, and it's all about zooming out sometimes to understand what is normal and what isn't. And it's very clear, and we also did this was on purpose. So during spring break, they discontinue the study, they go do whatever they do, and then come back to the study. And so there's that kind of lag. And you can see that they pick up just right where they left. And you can still tell that's the same individual, that's right where they were. And, and you can predict when there's menstruation, this is how it's going to shift, and then it's going to bounce back to where it was. You can see that. And you can also see, and we captured that in one of the participants and described that in the paper, but that when something changes, and that's what we're interested in. We're calling that shift. And this is another project that we're kind of working on. But there was a point in time, one of the participants, that during their menstruation, their profile changed. It's very abrupt and it's very obvious. You look at just these colors by colors, right? And you can see that something happened during that point in time where this participant went from a particular profile to something entirely different. And then it didn't bounce back. It just changed. And now that's the new normal for that participant. And we don't know why, we don't know what that means, right? But we do have all the data to kind of start to ask questions about that. Like, what does that mean? When somebody has a shift like that, something changed for the better or for the worse, I don't know, but something did change. And what caused that change? And what does that mean now for their health, for their, where does that, what changed, right? And we're very interested in kind of being able to understand, anticipate some of those shifts potentially, and intervene if that means a direction that is not beneficial, so that we could restore the balance to a healthy state. So, anyways, just very interested in understanding that it's kind of a test bed for all kinds of other things. We have some commitments with Wellesley to continue to follow with the Alumni Association, and throughout the years we continue to do these collections. We have done this now for 3 or 4 years and are continuing to do this. And it's not something that is really fundable from a, typical 4 or 5 year studies. These are studies that are discovery and they're long-term, and that are so needed. But unfortunately, our funding mechanisms aren't designed for this kind of thing. And what I find very, very, very sad, honestly, because it's so important, and these studies are very needed in a lot of areas for us to be able to understand. And I think sometimes, and I hope if there's anybody in a position that can maybe think about this and start these conversations, that science doesn't happen in cycles of 4 or 5 years, right? Sometimes maybe these studies should run for 10 years or more. And there should be funding mechanisms to support these kinds of things. We are very lucky to have had institutional commitments on both sides, and on occasion, we have benefactors that also are very generous and understand the importance of these studies. But it'd be great to have a general, scientific government to understand that as well. That some of these things just need time, and we need to understand them well to then be able to, better treat and address people's problems when they emerge.
Very interesting study, as were the others we've discussed. And one of the findings of the study suggested that exogenous hormones in contraceptives may affect vaginal microbiota. Can you talk about this a little and expand on this result?
Yeah, so one of the things we looked at was contraceptive methods. So we had participants that weren't using any, some that were using intrauterine devices, IUDs, or were using oral birth controls, systemic birth controls. And what we found was not necessarily, I guess, looking back, no, in hindsight, everything makes sense, I guess. But initially, I was thinking, that the systemic birth controls are going to be the ones that were going to be more disruptive to the microbiome. But what actually the data told us is that IUDs are actually far more disturbing to the microbiome. And, this is something we didn't quite capture as well as we are capturing now after seeing some of these results, because IUDs, there's many different types of those devices. And some of them are copper-based, and they have antimicrobial properties. So we think that might be what's behind some of this. It's not something we really anticipated to potentially be doing what we see, but yeah, we're seeing some disturbance to the microbiome with participants using those devices that we believe it doesn't necessarily, or maybe it does. Maybe it is linked to the hormones a little bit, but we are thinking it might have to do more with the copper-based ones that are having that antimicrobial effect. We don't know. We also have new data that we haven't published, but we are looking into the fungal microbiome as well, which we had only looked at the microbial side of these things. And now it's really interesting to also look at the combination of the bacteria and the fungus and how that gives us another insight into the ecosystem, which also contains a lot of the fungal elements that we were missing. So we just keep adding layers to the story, but I think it's really providing, again, that zoom out, understanding the whole story sometimes starts to, things that didn't seem to make sense then start to make a lot of sense. So it's giving us a lot of insights.
I can imagine that will be very impactful if more is learned. It's one of those things that would involve millions and millions of people, and you could imagine there's a substantial benefit possible.
Yep.
Yeah, incredibly impactful. And you write at the end of the paper that your study highlights the power and importance of longitudinal studies to detect critical patterns in vaginal microbial data. And the last sentence of the paper reads, and this is a quote, "The effort to understand the vaginal microbiome will ultimately facilitate discovery of new risk factors, diagnostic techniques, preventative measures, and treatments for vaginal microbial dysbiosis and other disorders related to women's health, including ovarian and endometrial cancer." So I'm just going to guess that you're hoping that this study and its findings will help motivate future investigations. Is that right?
Yes, very much so. So I do have some bias into the individualized medicine approaches. I'm a big fan of those to investigate complex and chronic conditions and also health in general, long-term things. I think for acute problems, the general standard approach, cross-sectional, is fine. But for something that has a lot of layers, is chronic, is complex, it's just, we need time, right? We need that dimension. It's very difficult to understand what is happening if you don't have at least picture frames of time sequence. Again, just going back to environmental roots and all of that, it's very difficult to put a story together when you just have fragments of different stories that aren't even connected to each other. It's a lot more meaningful when you have the story of even one individual through time. You can do it as an investigation, as a problem-solving type of investigation that is more meaningful in understanding how the different layers connect and what went wrong and when. So I'm a big proponent of longitudinal studies for these types of things. They, again, don't fit the standard funding model. They're very expensive, but what I think is that you don't need as many numbers as you do for cross-sectional stories, right? So cross-sectional, people enroll millions of people, sometimes, hundreds of thousands of people across the world to understand something, get some effect size. For longitudinal studies, you actually don't need as much of that. You just need, it's more of a understanding that particular individual really well and potentially their families and households. One thing that we are doing with our studies now is that we do household controls. So we're very interested in understanding, we have genetic controls in the sense of, people are genetically related to you is very important, but also people live with you. And to understand even the source of some of these microbes, for example, Porphyria monosamari in endometrial cancer, we don't really know where it comes from originally. It's not something very common in the environment. We don't know where exactly it enters the system and how widespread it is. Is it everybody in the household that now develops these things, or somebody you have an intimate relationship with? We don't really know. That's really important to know, right? These things. And so just even constructing studies through with the help of the IRB and everything to enroll even households is challenging currently, because like if I want to enroll a household of, say, somebody who is 40, 50 years old, and I want to enroll maybe a spouse, maybe minors that are living in the same household, maybe elderly folks that may not be able to provide written consent in some way, that potentially a vulnerable population. There's a lot of barriers to that right now that we have built that make it very difficult. It's to enroll families or enroll households like that in studies. But it's so important for us to understand the environmental components of these things. And I wish it was easier. And I hope it becomes easier as we see the value of these things. But it's kind of a barrier for these studies, both financially and just logistically. It really slows things down in a way that is detrimental to progress.
So, Marina, it's pretty obvious that you're passionate about advancing women's health worldwide. And another passion of yours is mentoring PhD students. So, you're the director of Mayo's Leadership and PhD program. And I understand that you've been quoted as saying this is the most rewarding part of your job. And I have to say, just listening to you and about your career trajectory, it makes a lot of sense. And it has to be really exciting to share that insight with students. So, tell us a little bit about this program and its goals, and also just what your role is in that program.
Yeah, so I do take a lot of joy from the students, and they, I also don't do science in the smartest or more effective way, and for warning to others, I do what I find fun and brings me joy in life. That's what I do. It's not necessarily the best career choices or or career progression. That's not what brings me joy. So what ends up happening is that my PhD students know this better than anybody else, but I give them the freedom that I was given as a student. I very much appreciated my mentors that did that to me, and I let them explore their motivations. I always tell them, it's your PhD. You're the ones going to suffer the most through this. So it should be something you're really dying to know and really want to know, because most days you're not going to find the answers. So, to keep it going, it's just got to be something you really, really want to do. And so I really force them almost in a way to follow their own ideas and their own, avenues. And I'm there to try to guide, but that's about it. And so they pull me in all kinds of directions, which is a little crazy, but it is fun in a way. And so the leadership program, it's a program that we started at Mayo as a result of something, we've been seeing. So, which is that at Mayo, there's great leadership programs and coaching that is provided to, typically faculty kind of, being promoted or that sort of thing, and kind of helps them do those jobs and perform those functions. Because as we all know, we as scientists aren't trained to, be PIs necessarily or lead others. We're trained to do science most of the time. And then the rest you kind of learn on the job typically, and some of us struggle more than others. We all struggle, but, we kind of just do the best we can. And I saw some of those programs, and, we were thinking there's a lot of folks, that never even get considered for leadership positions later in their career because they just never had good mentorship to lead them in that way. And then nobody ever really saw them as potential, having potential to be leaders. And it's too late at that point in time. So we thought, and this applies, of course, to a lot of less standard, I'll say, types of scientists, right? So we thought, why not provide this kind of, content and coaching and opportunities really early, like when they start their PhD, and just give them the knowledge of what's important for an efficient leader to, characteristics to develop, what's your default mode, kind of get to know yourself and how you can use your natural strengths and how you can grow and be aware of areas where it's more challenging for you. And everybody has their own areas. We all have areas we are good and struggle with. They may be different. And it's good to be aware of those and how to navigate that. And then how to understand other people, how to communicate effectively, how to have, what we call now crucial conversations, right? How to navigate challenges, because obviously it's not a matter of if, it's a matter of when, and it's often every day. There's disagreements and conflicts and things that are left unsaid and kind of brew in the corners and then explode. And how do you notice that? How do you manage that? How do you become a person that can help solve these for others, even and yourself? And that becomes very important in your scientific career, but also in your personal life. And it all comes together, right, into being just a good influence for others. I always tell students that no matter if you see yourself as a leader or not, you are seen as a leader by someone in your life. I guarantee that to everyone. Even if it's, a sibling or friend, a parent, even whatever, there's people looking up to you and kind of being influenced by you, whether you're aware or not. So might as well be aware of that and use that influence to do good things and have a good impact on others and be good role models too for other people. So I think that's just a very important part of life. And I hope that we as scientists understand that we have a lot of power because we have a lot of knowledge. And so we have a lot of power to influence our society and our communities, and we should use it for good causes and lead people as best we can.
So it's interesting to think that you arrived in the United States 20 years ago, roughly, to search for extraterrestrial life. And you've certainly had an interesting career path over these past 2 decades. So what advice do you have for young people who are pursuing careers in science? We always ask our guests to give some advice to young people.
Yeah, it's a tough question. I think the advice I have for, my students and students I come in contact with is to really doing what makes you happy. And that's the same advice I give my own kids. And I think that's the most important thing in life is to be happy and to enjoy what you're doing. If that's the case, everything else will fall in place. So find what is it that you're passionate about? What is it that you really want to study? And if your mind changes along the way, totally fine. There's no wrong in that. I think it's very important to pivot when you feel that the direction isn't taking you where you want to go. Just reframe. And we also have that in our leadership classes, actually, that that we pause to think about whether that's the direction we want to continue to go or not, and reassess. A lot of people often ask me about work-life balance and those sorts of things, and I tell them, it's always an ongoing thing. It's never the same every year. It's I have a certain plan, let's say, and then I reassess. And, it's important to pause and reassess whether you're happy where it's at, the balance, and if not, readjust, because life changes, things happen, you change, and so it's not always the same. It's going to change through time, and just be ready to adapt and find the right balance. So, that's my advice for younger scientists, I guess, is to not be afraid of that change, not be afraid to reassess. It's just like data, right? You reassess your conclusions every time you get new data. So you just pause and take a deep breath. It's worth to maintain your health, physical and mental, and continue to enjoy things. And I think it's really important. And I do this a lot, and maybe some people think it's a waste of time or whatever. To me, it's crucial. I take a lot of time to, spend with family, spend with my friends, and doing sports and things that really bring me joy and re-energize me, bring me energy. That's often when I have my best ideas. It's when I'm doing something completely unrelated. So those times are very important too, to recognize that that's part of re-energizing your brain and ideas and kind of recharge to continue to do good work and be able to take on adversity, because, scientific journey is a lot of adversity along the way. So you got to be accepting of that. And just keep going, and you'll do great things.
It's a nice answer, and we never used to hear the term work-life balance, and now I hear it way too much. And most people, when they bring it up, I look at them and think, yeah, I think it does need to be adjusted, but in the opposite direction you have in mind. So it's a cultural change that we're going through. But I really liked your answer. There was an older gentleman, a Nobel Prize winner, and he used to tell me that he had one big idea, some of which turned out not to be good, but they were interesting ideas, every time he walked to work. So he always walked to work. And as soon as he arrived at work, he would write it down, And in his very last years, we were walking from his house to work. And he walks very slowly at this point. And we finally get to work, and first thing, he opens his office door, he pulls out one of those long yellow sheets of paper, and he writes down his idea. And so he still had one idea each walk, but he walked slower and thought slower. So he maintained his one idea per walk, which I thought was a really wonderful thing. And it was great hearing him describe it because, as you said, you have many, most scientists have many of our best ideas not actually in the lab or sitting in front of the computer, but when you're doing something else. In his case, it was walking.
Right, because you're too focused. Yeah, I think that's the thing. To allow creativity to occur, your mind has to be relaxed. You have to be in a state where you just feel calm and relaxed to let ideas flow and connect. And if you're just really focused and really stressed, it's just going to be very difficult to get there.
I agree.
Yeah.
So, in terms of your interest, see, you knew we'd come back to this. In terms of your interest in extraterrestrial life, I have one last but long question for you. We interviewed Ed Weiler, who was the chief scientist behind the Hubble telescope and was director of NASA's Search for Alien Life program, among other things. He appeared on episode 148 of STEM Talk, and he believes that we'll be able to prove the existence of life in the universe off Earth in the next 20 to 30 years, sometime in that rough time range. But at episode 121 of STEM Talk, we interviewed Dr. Pascal Lee, a planetary scientist and director of NASA's Hot and Mars project at the NASA Ames Research Center. He said he thinks it's unlikely that other intelligent life exists in the universe, and if it does, we won't find it anytime soon. So Pascal's making the distinction between life and intelligent life. And Chris McKay is another person we interviewed on this topic, also from NASA Ames. And that also was a very interesting episode. So while we might find examples of microbial life on other planets, Pascal thinks that Earthlings are pretty much it when it comes to intelligent life. So, Marina, now that we've teed all this up, what are your thoughts on this?
Who do I want to get in trouble with? Is that what you're saying? Try not to throw. Well, I will say, I don't know if folks know David Greenspoon, but he wrote one of the best books, I believe, out there on these things called Lonely Planets. And he has a passage there that really resonated with me for this kind of answer. And it's going to connect to longitudinal studies and the concept of time, because I think I'm a little obsessed with this, but I think it's really, really important to think about this more than maybe we do. He has a passage there where he talks about how we on Earth and our solar system we are in a third cycle of our star, third generation. So our system has collapsed 3 times now, and every time the star collapses and the planetary system collapses, the next explosion. It creates a higher chemical complexity. So there's more chemicals that are more complex in nature are formed with each of these cycles. And we are in the third cycle, and we are one of the systems that has cycled the most, I guess. So he has an argument there that I find very interesting and unique, which is what he's saying is that, and I agree, that he believes that life is probably quite common. It's just statistically speaking, it's likely common, and intelligent life, obviously less common, but still fairly likely to exist. But what he said is that likelihood of those things happening probably increase with the complexity of those initial chemicals that are formed during this collapsing of the solar systems. And the cycle where we are now, we are one of the most complex cycles, or one of the earliest, I guess, probably life forms to emerge out of these kinds of recycling. And so there's likelihood that there's a lot of other life forms out there like us, but the likelihood that they are right next to us, or in a way that we can detect each other, statistically might be difficult in time, because we also have to coincide in that time, right, that we would be able to even establish contact. But as you go forward in time, and we're talking astronomical times, so not tomorrow, but as you go forward in time, the likelihood of life emerging and intelligent life developing increases substantially throughout those cycles. So his argument, which is interesting, is that maybe we are just one of the first, or one of the oldest things to emerge right now. But give it enough time, there's going to be a lot more emerging, and maybe we just came out too early or too soon to we're relatively alone or at least in the immediacy, potentially. But it was an argument I hadn't thought through before and thought in that way, but I thought it was very, very interesting. And it's all about, right, the likelihood of existing is very difficult from the likelihood of contact, right? And especially given the limitations we have currently in terms of space travel and detection mechanisms, which are just in the vicinity of our system. So that's a very different type of thing to answer in terms of will we find them or will they find us? It's different than them existing.
Yes, this whole discussion is interesting because any search for any form of life depends on a suitable definition of life, and in many respects, a useful, simple definition isn't readily available. We can understand life as we know it here on Earth and think about the requirements for that, and if we find liquid water or an energy source or some nutrients and a planetary environment with these characteristics along with molecular structures comparable to DNA and proteins, bang, we probably found life. But if it turns out that the fundamental characteristics of alien life are entirely different than life on Earth, it'll be really hard for us, I think, to notice it or to consider it as life. But I agree with you that the likelihood of life in the universe, just statistically, looking at the size, is highly likely, I believe. I'm much more pessimistic about intelligent life, just given how long it took it to develop on Earth, but for other reasons too. And then finally, even more pessimistic on intelligent life with that we might be able to communicate with. And that seems to me vanishingly unlikely. So for me, in the Drake equation, n equals 1 or very close to 1, like 1.2 or something. But I love that. I hadn't heard about the book and about this gentleman's perspective on it. It's certainly novel and very creative.
Yeah, and very interesting. Maybe, if our challenge is to stick around for long enough to see that bloom.
Well, yeah. If Earth is an example, it took a very long time for intelligent life here.
It took a very short time for life itself, though.
That's right. That's the distinction. And that's why I'm very optimistic about one and less optimistic about the other.
I mean, if you consider us intelligent, I guess, but that's another question.
Yes. And that seems increasingly in doubt.
Spectrum, yes. Well, Marina, thank you so much for joining us today. This has been a fantastic discussion, and we've really enjoyed hearing about the work that you've been doing and continue to do. So thank you so much for joining us.
Thank you. Appreciate it.
Yeah, you were great. Thank you very much.
STEM Talk. STEM Talk. STEM Talk. STEM Talk. STEM Talk.
So this is such a fascinating discussion and such exciting work by Marina, and it's so encouraging to think that the microbiome has the potential to help us detect such things as ovarian cancer, even at an early stage. But it's particularly encouraging that we may be able to use the microbiome to predict the patient's response to cancer treatments as well. So Ken, I really think Marina is someone whose future research is something that we really need to pay close attention to.
Agreed, Don. I suspect Marina and her colleagues are on the cusp of developing new ways to assess and manipulate the microbiome to both detect and treat disease. This is exciting because the microbiome is still very much understudied in my view, and it's a critically important component of human health. So stay tuned. We're likely to be hearing much more about this exciting area of research in the future.
This is Don Carnegas signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.